Hello everyone, today we're going to talk about Lab 5, which is Affinity Chromatography. And so by the end of this lab, you should be able to describe the principle of affinity chromatography and predict where a protein will elute. Given a particular protein, select an appropriate affinity chromatography medium if it's applicable to that protein. And then finally, select an appropriate elution method for the enzyme affinity column chosen. All right, so just an overview of the technique. Affinity chromatography separates proteins based on their ability to bind a ligand. And by a ligand, this can be something like a substrate. So for LDH, this would be lactate or pyruvate, or something like a cofactor. So for LDH, this would be NADH or NAD+. And so this type of chromatography is a very specific chromatography. And so it allows us to effectively pinpoint one protein, or at least a very narrow group, a very small group of proteins. And that's just going to depend on how general or how specific the ligand that you're using to purify is. So if we look back on the way that we've purified our protein, you'll notice that we've gotten that as the purification procedure has progressed, the techniques that we have chosen have gotten more and more specific. So when we started with ammonium sulfate, we were able to eliminate a broad swath of proteins, but there's also going to be a lot of proteins, other proteins in that 65% cut that we took. So this is a very broad technique. Then we go with ion exchange chromatography. And this is a little more specific, so we select out the proteins that may have had a similar solubility, but they had a different charge. But then finally, when we get to affinity, ideally we're selecting for only LDH, but while this is a more specific technique because this is a more narrow subset of proteins, because it's the proteins that were the same hydrophilicity, a similar hydrophobicity, a similar charge, and have a similar affinity. So in this case, binding nucleotides like NADH. But the pool gets smaller and smaller, and that'll become more apparent when we run our SDS page gel in lab 9 and 10. So the idea is as we're progressing, we're becoming more specific, and affinity chromatography is the most specific step that we use. So there are two types of affinity columns that are commonly used. And the first is a resin. And so this is where the structure of the column material actually mimics a particular ligand. And so an example is Cybacron blue, which is actually the resin that we're going to use in this lab. And what Cybacron blue does is it actually mimics nucleotide cofactors, such as the NADH cofactors and FAD. And all proteins that bind that specific nucleotide as a cofactor are going to also stick to the resin. So if we've got proteins coming out in the same IEX fraction that also bind nucleotides like NAD or FAD, they're also going to co-elute and come out with there's a good chance that they will come off at the same time as are in the same fractions as LDH. So we've got to make sure we're careful. Although ideally that wouldn't happen. But if you look at the structure of Cybacron blue here over on the left, you can see there's that 3-ring structure that we saw that we'd see with FAD, And then we have the single rings that mimic a nicotinamide ring and NAD or NAD plus. NAD, NADP, and then up here that mimics FAD, that flavin nucleotide there. So the second, and again, so this resin is the type that we're going to be using in this lab. But it's worth mentioning another type of affinity chromatography media, and that's a type of column where the ligand is actually attached to the resin. And this is a very common form of column chromatography. We use one of these types in the other, in 370L, and some of you may have used them in your own research labs. If you've done research, I know we use the first example in my lab. So this is a nickel NTA affinity column. And in this particular column, you have a bead, and the bead has this linker that's terminated in an NTA group, and those NTA groups coordinate a nickel 2 plus ion. And so 4 of those coordination sites are taken up by the NTA molecule, but there's 2 sites left on the nickel column that can coordinate other molecules. And so what ends up coordinating these molecules are proteins that have a 6x his tag. So if you have your protein sequence, and then you end with literally 6 H's. And so if you're overexpressing a protein in the lab, you can insert 6 H's to either the C terminal or the N terminal into the protein. And when you run it over a nickel affinity column, the lone pair electrons, now you have to make sure it's at the right pH so your histidines are deprotonated. So just like the catalytic histidine in LDH, you have to make sure those histidines are deprotonated. But if at the right pH, they can coordinate the nickel ions, and then the his tag will stick to the column. And so again, but one of the pitfalls of this method is that proteins that have a lot of histidine or like a cluster of histidines can also bind to the protein, can also bind to the column. So depending on the mix of proteins you have in your slurry at the beginning of the point, you can either get a really pure sample, like a 90 to 95% pure sample, but you can also get a lot of other proteins stuck in there, so you have to be careful. Another type of common ligand-based affinity chromatography is an antibody column. And so in this case, you take an antibody that binds to your protein of interest, you covalently link it to a column, and so then when you flow your protein, when you flow your protein through, your protein will stick. Or you can have a column where you put some kind of an antigen on the column that your particular antibody that you want to purify is attracted to, and you can use either of those methods to um, exploit the specific binding of antibodies and antigens. So that's one thing you can do, depending on your protein, if you have like a very unique protein. But this can get expensive because antibodies are not cheap to make, depending on the particular, and depending if your antibody is commercially available or if you have to have it specially made. And then I wanted to give just another example of affinity chromatography where there's a ligand tied to the resin. And this is an example that I've used in my research. And this molecule here is called methotrexate. And it's actually an inhibitor. And it's actually an inhibitor of an enzyme called DHFR, or dihydrofolate reductase. And so what happens is you covalently link the inhibitor of an enzyme to resin bead, and then the enzyme will bind to its inhibitor, and that's how it will stick to the column. So then the question is, so I showed you all these different ways that we can get our protein or enzyme of interest to stick to the column, but how do we get it off the column? And there are several different methods that we can take. The first and perhaps the most obvious method is using a ligand gradient or competitor binding. And so what happens is once you have your protein stuck, so the purple guy there is stuck to, so if this is LDH, for instance, and it's stuck to the nucleotide cybercron blue resin, one of the things we can do is we can load on the column, and then we can wash the column with either a ligand, with either a large amount of the ligand or competitor binding. And so what we do is we overwhelm the column with free inhibitor. So what happens then is the concentration of ligand that's free is going to be much, much greater than the concentration of ligand on the column. So instead, the protein is going to grab on to the solution ligand, and then it's going to come off the column. So you can see here, if before LDH was bound to the cybercron blue column, but once we were to titrate in free NAD plus, LDH, and if we added enough NAD plus, so like NAD plus in solution was much, much greater than the cybercron blue column, then it would prefer to come out of to bind to the larger amount of the ligand in solution and then come off the column. However, this method is not perfect, and there are several concerns. The first is the cost. So I looked it up, and the cost of NAD plus is $200 for 5 grams. Now, this is a teaching lab, and so we need to make sure we have the materials we have. However, we also need to be frugal and careful with our money. And making an elution buffer, and typically for an elution buffer, you need like 1 molar concentration. And for our column, you're going to need like 20 to 50 mils, depending. That's not economical at all. That's just a lot of money. The second concern with substrate or like ligand titrations or method of elution, once you have your protein in the solution, it's already bound to the substrate. And so if you want to do Michaelis-Menten kinetics, it's going to be messed up because your enzyme is already bound to a ligand. So it's going to throw off your measurements. So if you want to elute this way and then you want to do Michaelis-Menten kinetics, you have to unfold your protein and then refold the protein. And this brings up several problems. First things first, it takes a lot of time, and you have to unfold the protein using urea or guanidinium chloride. And sometimes when you unfold a protein, if you have a very large or complicated protein, there's a good chance that not all of your protein is going to refold properly, and that can also mess up your enzyme concentrations and the activity of your enzyme. So there's a lot of extra hassle that's involved with this step. The second method of elution is a pH gradient. And so what happens is if you change the pH of the solution, you're going to change the charge of the protein. And if you change the charge of the protein, then that might disrupt some of the protein ligand interactions. And we cause it to fall off the column. However, there are also several concerns with this method, and the first would be denaturation. So, how far do you have to change the pH in order to get your protein to change its charge? And is your protein active or even folded at that concentration? So you run the risk of denaturing your protein on the column, which isn't good. Also, for many enzymes, activity is pH dependent. And so you just don't want to mess with the activity of the enzyme when you're trying to purify it. That's just not a very good or very smart idea if you can avoid it. And finally, the last method, and this is the method that we're actually going to use during the lab. And the idea behind using a salt gradient is that if you have high enough salt concentrations, um, you will mess with the ionization of the protein a little bit. So you may be masking some of the charges, but ultimately the presence of the salt is going to disrupt the protein ligand interactions. But unlike pH, where you would be messing with the ionization states of the amino acid side chains on the protein, you would just add salt and it would mess with some of the interactions by the ion bonding somewhere instead of an amino acid. So you'd be messing with the interactions, but without having to worry about messing with the structure of the protein. Now, one concern with this guy is you will have to dialyze. So you're going to have to get rid of that salt, some of that salt, because chances are your enzyme isn't going to be active at the high concentration of salt that you're going to have to elute. So you are going to have to add a dialysis step where you dialyze it into a low salt buffer. However, this is still the cheapest and easiest method, so it's the most cost efficient. You get the biggest bang for your buck, but ultimately it's of minimal harm to the proteins. So it's the safest and the cheapest way for our proteins is to use this method. So now I'd like you to take a minute and think about two important questions. First is why do we dialyze our sample before the affinity column? So we talked about why we wanted to get rid of any salt before we put our sample on an ion affinity column. It makes sense because the salt might bind to the column instead of the protein. But why would this matter for an affinity column? I want you to pause the video and think about it for a minute. So we're exploiting the ability of salt to disrupt protein ligand interactions to elute the column. So if we have high salt concentrations, then that might interfere with how the enzyme binds to the column. And I mean, we could test it and find out for ourselves, but it's better to not take a chance. The second question is why will we store our sample in the fridge, not the freezer? So, in particular, we're really concerned about preserving our enzyme's activity. So, why is it better to put it in the fridge, not the freezer? So, every time you freeze, so taking a sample in and out of the freezer is called a freeze thaw. And so, every time that you freeze thaw an enzyme sample, some denaturation is going to occur. And so if you freeze thaw, obviously we don't want to lose protein, but as you freeze thaw the enzyme, when you're going through multiple purification steps, if you lose some protein along the way, it's not that big of a deal. But on the last step of our purification, we want to make sure that all of our sample um, is folded and active. So we don't want to do anything that would make the protein angry or denature it. So just speaking about some of the logistics of this lab, in terms of how the column is set up and how you're going to collect fractions and record your data, it's virtually identical to the ion exchange lab. So I don't feel a need to mention that again. We'll cover that at the beginning of lab. But just a really quick run over of the things we're going to need for the lab report. The first thing first is you're going to need to measure the volume of your pre-affinity column sample. You're going to need to run 2 activity assays on the pre-column sample. Then you'll measure the volume of your 2 to 3 most active fractions, and then run 2 activity assays on those post-column activity or those active fractions. And before you leave, so actually after you run that, I measure the volume, and then you're going to save 250 microliters because you're going to need 250 microliters of both your pre and post column sample. So I know a lot of people had problems with that back in lab 3. So we got to make sure you save 250 microliters of pre and post. You put them in little Appendorf tubes. You give them to me or Lindsay. We'll put them in the box. They'll go in the freezer because we're going to use them for analysis later. And then finally, give your 2 most active fractions to Lindsay. Nat's the TA for 370L this semester. So pour your 2 to 3 most active fractions together, and then after you've taken 250 microliters and after you've assayed the activity, give them to Lindsay. She'll dialyze them, and then again, as always, check with Lindsay or myself before throwing anything out. And if you have any questions, please feel free to set up an appointment or visit me during my office hours.